Okay, well, I struggled a little bit on what I'd like to talk about today, and so I decided I'd sort of meander about the idea of trying to get a drug approved and how one can do that through the academic means. Although maybe my talk would have been better before the subsequent preceding talk, because it might be more relevant in that way. But here are my disclosures, some of which are relevant. Okay, I'm going to split it up into a mini introduction to PSMA, which I think probably has already been done well, and then we'll talk about the data we have and the writing of an NDA, the struggles with that, and then talk a little bit about how academic literature is used to support an NDA, and talk about how it's important that our literature is improved a little bit. And then finally, a couple comments on outcomes and what's going on in that setting that I'm aware of. So mini introduction to PSMA. We know about gallium-68 PSMA, right? It has this binding site that looks like urea, and it binds to the PSA molecule. It binds to the extracellular domain, and it's been used extensively, and initially back in the day was shown to be superior to fluorocholine in Australia and Germany. Now, this is a case that's a classic PSMA PET case, and what we like about PSMA is that you can see both bone lesions as well as soft tissue lesions, right? So hopefully, not far in the future, we can get rid of the bone scan and have a single imaging study that we can do both bone and soft tissue staging in a patient, which will become much more consistent as we move forward. So that's why we like it. Now, we all know that there's about 1 million of these compounds, and I think it's a word jumble. There's so many of them. The vast majority of them have the same binding moiety, and so really it's the chelator that's varying that does change its biodistribution, which can affect the sensitivity. With, if you have a larger blood pool, like some of the earlier compounds, that might lead to a lower detection sensitivity, and many of these are now in clinical trials. So at UCSF, we use PSMA-11 solely because it was not patented, it was available. Doesn't mean that it's any better or worse than these other compounds, and we are very much so looking forward to many of these compounds getting approval in the United States, including, for example, the bottom left there, GCF-PYL is in phase 3 trials, and hopefully at some time will be approved. But PSMA-11 is the one that we could move forward with in academia because it's not patented and not run by a company. So, we did do some stuff and collaborate with UCLA. I just wanted to show this slide of PSMA versus fluciclovine. And I think everyone can agree that fluciclovine can be improved upon. And I think this data sort of highlights that. The UCLA group did this head-to-head study in 50 patients. Patients who were getting standard clinical care of fluciclovines were enrolled before the fluciclovine to get a PSMA PET within 7 days of getting their fluciclovine PET to do sort of a paired evaluation. And the take-home point here is that with PSMA, you obviously see a lot more nodal disease than with fluciclovine, and a lot more distant metastatic disease. Okay, and this was probably true for all PSMA compounds out there, and so this is why we're all very excited about having a new compound moving forward, and we need to get that approved by the FDA so we can have more widespread use. I think something we don't talk a lot about in PSMA literature is the idea of inter-reader agreement. A lot of the literature out there focuses on detection rate. We talk, every slide is PSA less than this, greater than that. This is how many patients we see disease in. And if you look at the literature, it's always with a single reader. And when you use a single reader, your detection rate sort of creeps up because you're incentivized to overcall disease. And so it's really important when you think about how to do these studies to have multiple blinded readers, readers who aren't from your institution, who haven't seen those studies before, who don't know the pathology or any of the outcomes in that patient, to actually truly evaluate the characteristics of the imaging study to detect disease. So in this example for here, this is FACBC or fluciclovine, PSMA. You can see in the tumor, you have a fairly similar multi-reader. There's a Fleiss kappa with 3 readers, but you can see when you get to the nodes, you have a very overall much lower inter-reader variability with fluciclovine compared to PSMA, and I think that's really important. It's also really important when you want to sort of report detection rates to have more than one reader, because you're going to have to agree upon it. When you have this, they come to consensus. That's not what I mean. I mean, you blind read separately, and then you can have a consensus read from the blinded readers, but you can't have just a group of people evaluate studies and report a detection rate, because it does, in both with PSMA and every other imaging study, results in a over-representation of the detection rate in that disease. So I think that's sort of, the FDA forced us to do that in our pre-IND meetings, and I was sort of like, I didn't really appreciate how important that is to actually get usable data. So let's talk about an NDA. So I was very lucky to collaborate with Johannes Thomas Aaron in the UCLA group, and there is a whole ton of people involved in this, but Xiao Zhu, who is a regulatory expert at UCLA, was incredibly important. And at our site, Ashley Michaud's our radiopharmacist who runs the cyclotron facility, and then there's probably 30 people, 40 people total who help bring all this data together to make it work with an NDA. Now, this is us. I always like to show this picture. This is us at the FDA last August at our pre-NDA meeting, and it took us a year to get this. It was a year and 4 days from our pre-NDA meeting to get our NDA submitted, which we finally submitted last month. And I just want to go through a little bit about what an NDA looks like. So for those of you who don't submit NDAs daily, this is what a general overview of an NDA looks like. So we have Module 1, right, which is Module 1, I can go back, whatever. You have your package insert, and then it's actually sort of funny. Module 1 is mainly miscellaneous documents, letters of reference, and things like that. Module 2 is an overview of Module 3, 4, and 5, however weird that is. Module 3 is your CMC section, so you have to put in, chemistry and manufacturing. 4 is toxicology, and I should acknowledge over there is Wolfgang Weber. Our toxicology data came from the Technical University of Munich, who had done this because they were running a prospective trial in Germany, and they needed this small animal tox data. Without that, we wouldn't have been able to really fill out Module 4 effectively. So it took a lot of the data, and you'll see a lot of the data in our NDA wasn't data that we acquired. And then Module 5 is the clinical data reports. So the clinical study reports or data reports is really the core of your NDA. So Module 3 in our CMC, I just want to make one comment about that, which is an important thing, is that you can actually make gallium-68 on a cyclotron. And so our NDA is the first NDA to have gallium produced off a cyclotron in the NDA. And I think it's an important caveat because one of the issues here is we're not going to have enough gallium generators at all, not anywhere near enough gallium generators to provide gallium PSMA for anywhere number of prostate patients there are. So having the ability to make gallium PSMA on your cyclotron is important. There's a lot. Right now, our final product's about 42 to 45 millicuries of gallium PSMA, so maybe you can get 5 doses per synthesis, but soon people will be developing, and they're currently developing, solid targetry, where you can make much more final product and actually maybe make enough doses to get 15, 20 patients out of a synthesis, if you have enough PET CTs to image the patients on. Obviously, that's sort of a PET CT bandwidth. But this is a really important aspect of making this available. And a lot of work, not from our site, but out of collaborators from Iowa, Michigan, and Mayo Clinic here to how to do this, how to write the NDA, educate the FDA about what cyclotron-produced gallium was really critical to make this happen. And actually most of this work wasn't done within our institution. It was just collaborating with a bunch of other institutions who laid the groundwork for this. So Module 5 is really where the majority of the work is. And so the basis of our Module 5 was 4 clinical study reports. 2 were meta-analyses, and 2 were clinical data from our prospective studies, the largest of which was our BCR clinical trial data, which was published this year with Wolfgang Fendler, who was the first author on it. And this was data pooled from UCLA and UCSF with 635 patients. The median PSA was 2, so a fairly low PSA. We had 9 blinded readers. There was so many studies that we actually broke them into groups of 3. So we had 3 composite blinded readers on all the study. We had 223 patients with a composite validation. And this is the really big issue with this, is you can't biopsy these patients because the lesions are so small. It becomes very difficult to actually have a histologic confirmation in all your studies. And the FDA is obviously very interested in patients with a PSA less than 2. And so it becomes very difficult to actually have this composite endpoint. So we did a composite endpoint combining both histopathology and then this sort of very unwieldy imaging follow-up. Did the patient get external beam radiation therapy and the lesion get smaller? Did a sclerotic lesion develop after starting ADT? And we had 4 pages worth of different ifs to determine whether or not lesions were true. And so we had 223 patients who had a composite endpoint, and the positive predictive value for the composite endpoint was 0.92. So reasonably good and consistent with the literature. The thing I do like about this is our inter-reader variability. None of these readers, the 9 readers, actually were even from the United States. None of them had seen the studies before, and you can see our inter-reader kappas were, for everything outside of the prostate, in the 0.7 range, which is actually very good for an imaging study. And so this is a fairly reproducible imaging study, which I think is key. The background activity is so low that when you see a lesion, you're fairly confident that that lesion's real. So I think that's a really important test characteristic that we need to think of moving forward. Now, the other half of our clinical study report was a meta-analysis, and so I just threw up a slide here on the left of the meta-analysis of the pre-radical prostatectomy data in the literature, which in all honesty, there's very little good literature. One of the best is out of the Technical University of Munich. And then this on the right is sort of the positive predictive value pooled across all the patients that were reported to have histopathology in the literature. The take-home point here is our literature is incredibly poor. If you go back through and try to do a meta-analysis of the literature, there's very little literature that does any attempt to have a correlation to a composite endpoint, pathology. There's no blinded readers. There's very little description on technique. And it actually really limits our ability to pool that data to support subsequent submissions to the FDA. And I think that my learning lesson from this is that we need to be in academia. Industry does this well because they're all designing studies for approvals of drug. But academia does not design imaging studies in a way that's poolable or usable for an NDA. And I think that's the biggest take-home point from my experience through this, is that we need to rethink how we design imaging studies to be something that could be incorporated into subsequent submissions to the FDA, particularly when you're working with a novel imaging agent. There are thousands of articles on PSMA PET out there. Very few of them even report detection rates that's usable, let alone have blinded readers. Like, none of them have blinded readers nearly. And it makes it very hard to have any usable data out of our literature. So I think that's one of the most important take-home points. So quick comment on outcomes, which I think is important. So this is data out of UCSF. In essence, everyone who does PSMA PET has these same papers because they're all the most obvious things you're going to do. So Australia has these, we have these, UCLA has them, Germany has them. They all say the exact same thing. But what we did, just like everyone else, is we looked at patients who had a PSA less than 2 after radical prostatectomy, standard of care therapy or salvage radiotherapy, plus or minus pelvic nodes, so you do prostate bed. And at UCSF, you'll get pelvic sidewall nodes. If you look in these 125 patients, 53% of them had PSMA positive disease, and 30% of them had disease that would be missed by standard RT fields. And standard at UCSF includes the pelvic sidewalls because we have Mac Roach. And so that is really interesting, because if you look at the percent of patients who recur 2 years out from salvage radiotherapy, it's 30%. So it sort of makes obvious sense that if you could tell where that 30% of disease is, you could potentially cure those patients. And I think that understanding is what led UCLA to start this trial. And I think this should get a lot of credit to have an academic site fund their own prospective randomized trial of local targeted, like in essence the STOMP trial, right? Targeted radiation therapy to PSMA positive lesions in patients post-radical prostatectomy with a PSA less than 2 with a 1-to-1 randomization. So I think it's really important, this type of study, to get published before the availability of these drugs, because once these imaging agents are available, we'll never be able to run this type of randomized study. And in essence, we've talked about this endlessly in cooperative group settings and other settings, and we never actually get a trial. And once this imaging agent becomes available, you'll never be able to really randomize again. So it's critical for this type of study to get completed before the availability of these agents. And I'm really, I'm proud to participate in this study, and also that someone is funding this on their own because they know the clinical need to have this type of data out there. So, in summary, PSMA PET is superior to existing radiotracers for the detection of metastatic prostate cancer. We need to improve the quality of our imaging literature to support future work, and I think that's really one of the most important take-home points because of the importance of meta-analyses and submissions to the FDA. And the next step in understanding impact of PSMA PET on patient outcomes. We have to focus on that, particularly before this grows like wildfire and is used by everyone in the United States. So thank you very much for everyone and the huge amount of people at UCSF and UCLA and all the collaborators other institutions who helped make this possible to actually submit an NDA for this drug. Thank you.